And then we'll get going.
Okay. Dr. Vaasjo is originally from the island of Puerto Rico and trained at the UPR-Institute of Neurobiology, studying sea-slug neural circuit evolution. Later, he transitioned to the University of Chicago, focusing on the basal ganglia and dopaminergic circuits in mice. Currently, he just completed his Ph.D. in the Galazo Lab at Tulane University and is now a postdoctoral researcher in the Chamberlain Lab at the University of Pittsburgh. He was previously a visiting researcher at the Woods Hole Marine Biological Laboratory, an SfN-NSP Fellow, and was recently awarded the NIH-DSPAN F99 K00. He aims to become a benevolent independent investigator within 3 to 5 years.
Welcome, Lee. Thank you.
Thank you. We'll update that to be between 2 to 4.
There we go. There we go.
All right. Yeah, thank you for that introduction. So why don't I just sort of get to it? Let's see if this will work. Reconstructions, launch meeting, just screen, share. Is this working?
Yep,
Yes.
that's working.
All right. I can't see the top. How can I move this around? Okay, and I'll see if I can move this maybe down here. All righty. Well, again, thank you very much for that introduction. And so because of what we're doing today, I just wanted to give sort of a brief background of where I'm from. You know, I didn't start out being some circuit mapping neurophysiologist. So let's just sort of see how I ended up where I am, and then we'll go into some more deep science. Okay. Right. So in Puerto Rico, I originally trained as a marine ecologist, and that's what I wanted to do, you know, for the vast majority of my life. And I had a lot of research experiences, including coral aquaculture, counting lionfish, and working with sea turtles. And that was just really over the moon. It was a really incredible experience. But it just left me with this gut, you know, how are these animals doing these incredible behaviors? How are they interacting with the environment? So when I had the opportunity to start working in the neuroscience of marine animals, it really is a transformative experience. That's where I joined this NSF-URM scholarship. Okay, so then I graduated from the University of Puerto Rico. Later I went to, then I was running a microscope core facility there. And I said, look, you know, sea creature, this is fantastic. I'm having a great time, but the tools are limited, and this isn't the only, you know, mammals are just so different, and there's all these genetic tools, and I just need to diversify. And it was something that really a number of mentors had pointed out, like, hey, you know, you lack some really critical skills that will take you to the next level. You know, work in a different animal and then come back or do whatever you want. So I decided to work in transgenic mice, and that's where I joined the University of Chicago, joined Jeyifous Lab. From there, I was a tech for 2 years and then went and did my Ph.D. at Tulane. Why Tulane University? You know, I wanted to go to a city where I had family. It was a nice, fun city. And I said, look, a PhD is hard enough. Let me go somewhere that's like the background is fun and I can just go do fun stuff when I'm doing my work hard, play hard PhD life. That worked out really, really, really well. I had joined the Galazo Lab, and really it was a productive PhD. I joined, you know, I became an SfN-NSP. Then I won this NIH F99 K00, which at the time was hyper competitive. This is Stanford, Yale, Harvard, all of these super high level people, 15, maybe 12 to 15 people get this. And to be from Tulane and getting it was a really big deal at the time. So that was really cool. Then I decided to do my postdoc at the University of Pittsburgh. So why am I in Pittsburgh instead of, like, you know, my Stanford UCSF offers? Sort of I get this question from more senior people. Why am I in Pittsburgh? You know, here I could buy a house. I'm doing the exact same incredible science that I could do. I'm doing the same incredible science I could do at Stanford or at Yale. I have the same microscopes, the same equipment. I'm just in an affordable location. So it's a really big deal. You know, we're planning for a family. It's an opportunity that you wouldn't get if I was in San Francisco with like a $5,000 a month rent. So yeah, I'm paying $2,000 for a mortgage of my own house. So pretty good. Anyway, so life decisions, right? It's sort of where does the science lead you, but can you also have a really great life? And that's sort of what I've worked on. Anyway, so now let's talk about the science. If you have any other questions about sort of work-life balance, you can ask me that as well. All right, so we're talking about the development and physiology of dendrites. Really, today's talk is focusing on pyramidal neurons. I've worked across different neuronal subtypes, but we'll focus today on pyramidal cells. Really, I didn't even know how to name this talk because it's so broad about dendrites and their development and their function and their synapses. So we'll just sort of encompass it in this title, and then we'll see where it goes. All right, and so all right. So, you know, neurons come in so many, just tens of thousands of flavors, right? And we'll be focusing primarily on pyramidal cells today. Can you see my mouse, or should I put a pointer on? Not a pen. Let's put the pointer on. Okay, can you see the pointer?
Yeah.
Cool, thank you. So we'll be focusing primarily on pyramidal cells. This first part of the talk is in the cerebral cortex. And so if you're engaging in this meeting today, you're definitely engaging your cortex. It's just fundamental for information processing and then motor output if you're taking notes or doing whatever you're doing. Oh, then I'll use this. Okay, and so if you hone in and zoom into the cortex, here's just a camera lucida drawing from Ramon Cajal. You can see these pyramidal cells. They look like little pyramids, and they have these large dendrites. Okay, just beautiful dendritic arbors. Here, one's in the deeper layers of the cortex, projecting to the upper layers of the cortex. So this is the surface of the brain, and this is deeper layers of the cortex. So one of the first things you can see in this image is that cells in different layers and sort of different located cells have different dendritic arbors. And I found this really fascinating when I started my PhD. Was like, well, what drives these different dendritic morphologies? It's just sort of a big deal. And you can see the cell body's this tiny thing, and the dendrite is really, it's like a millimeter long. It's a big thing. And just for reference, this is the primary site of receiving information for the neuron, right? So its antenna, if in a way, is just shaped in all these different ways. So how are these different dendritic arbors generated? So to understand how are these dendritic arbors generated, we have to go into sort of a molecular perspective of what are the genetic mechanisms that guide the development of these cells. So if we look at a cortex, this is a mouse cortex. Here are a number of neuronal markers that are really, you can see, are quite specific to different layers of the cortex. And so these are primarily transcription factors that guide the development of cell identity. And so then the cell identity will decide, okay, well, I'm a pyramidal cell, I should have a dendrite like this, or I'm a layer 6 pyramidal cell and I'll have a different sort of morphology. We'll get into these specifics right now, actually. So just a little bit of this sort of moved in a weird way. Anyway, this decided to not save my modification. That's really too bad. Okay, so today's talk is split into two. I have other stuff if we have time, but we'll talk about the identity maintenance and how I was reprogramming the identity of cells. Basically, when I'm changing the identity of one cell to another, I found that I could also change their dendritic arbor, and that's really a big deal in the field. The second part is what I'm doing in my postdoc, where I'm looking at how inhibition, like inhibitory neurons synapsing on the dendrites, are able to modify these large calcium currents. And we'll see how this relates to dendrites in a minute. Oh, get it and make it a little bit larger. Too many screens. Here we go. Okay, so I was commenting about this sort of cell identity element, and well, how do we as scientists define different cell types, right? We might have circuit connectivity, right? Where do these neurons send their axon to? That's a big deal in defining cell type identity. For me, a really big deal, and for a lot of scientists, is the dendrite morphology. Here, we're only looking at excitatory cells, pyramidal cells being a type of excitatory cell, of cells across layers of the cortex. And you can see from the schematic that the dendrites are really different, right? You might have cells in these deeper layers projecting to the surface, or cells also in the deeper layer projecting sort of to the upper layer 5 or something like that. So that was, different dendritic morphology. Another way we define cell types is through electrophysiological, right? Their biophysical properties. Also, where are they even just located in the layer of the cortex? This sort of helps us define their cell identity. Another big deal that I don't think I'm talking about today is about the birth of these cells, like the day they're born during your development, right? Neurons born earlier versus neurons born later will have different cell identities for the most part. Not every rule is set in stone in development. And a huge deal is, well, what is their molecular makeup? What about the genes that these cells express is a huge deal in defining and identifying the cell type identity. So the question is, how does gene expression define neuron identity and dendritic morphology? This is one of the sub-questions during my PhD, just one of like 25 questions. Anyway, but let's talk about the main types of excitatory, like pyramidal excitatory neurons that we'll be talking about in this part of the talk. All right, so the cortex is a layered structure. We have layers 1 through 6, then with the white matter down here. And if we take a coronal brain section of a mouse brain, we can see in the upper layers of the cortex, layer 2 through, we have neurons that project their axon to the contralateral hemisphere. These are callosal or cortical projection neurons. And then if we go to the deeper layers of the cortex and take a sagittal cut of the brain, we'll see that we have in layer 5, neurons here project primarily to the brainstem and even to the spinal cord. So in humans, you'll have axons that are even a couple feet long, right? So axons being a key defining element of a cell type. Okay. Oh, and these are cells primarily affected in ALS. In layer 6, in this one of the deepest layers of the cortex, we have neurons that project primarily to the thalamus. And this is really critical. These cells are fundamental to sensory motor integration. So a little bit about the development of these cells. It is that these two cell types, the layer 5 and the layer 6 cells, are really lineage related. So they come from the same progenitor cells during development. Also, they have a lot of genes that they share in common that they express. A lot of these are transcription factors. However, transcription factors and regulators of transcription factors, these transcriptional regulators. However, if we start taking a couple of these differentially expressed genes, or DEGs, differentially expressed genes, and manipulate them in different cell types, we start to see sort of what happens to the cell when we're manipulating these key differentially expressed genes. So for example, if we overexpress FESF2 in the deeper layers of the cortex, cells tend to become, during development, they tend to become these layer 5 subcerebral projection neurons. If you knock out this gene TBR1, they also become layer 5 subcerebral projection neurons. It's down here. Okay, so then we found that this TLE4 gene had sort of a similar probability that it did something like this. No one had studied TLE4 what it did, and that was, well, hey, pick this gene out of a hat, see what it does, and I'm quite lucky it did something pretty cool. All right. Cool. So a little bit about TLE4 and its function during development is that when it's expressed in layer 6 cortical thalamic neurons tend to become, going back, when TLE4 is expressed during development in these undifferentiated postmitotic cells tend to become cortical thalamic. And they're also suppressing the subcerebral projection neuron lineage. However, when TLE4 isn't expressed, they tend to become these layer 5 subcerebral projection neuron or upper motor neurons. Okay, so then the paper that defined the work that I'll be talking about today was this work by my PI when she was a postdoc, which that she found that during development, really early in development, when you knocked out TLE4, FESEV2, that gene, it's upregulated, and neurons tend to become these layer 5 subcerebral projection neurons and develop an axon that projects into the brainstem. However, I was shown this was a preprint at the time, and I'm shown this data. I'm like, oh, wow, this is cool. Well, cells now become layer 5 subcerebral projection neurons. The preliminary data show that it also changed its dendrite from the sort of squiggly little dendrite that projected to like layer 4, the upper layers of layer 2 of layer 5, versus this layer 1 projecting large dendrite. Like, well, okay, this is pretty cool. But then I saw that FESEV2 is actually constitutively expressed throughout life. Like, why is this gene that's essential during development expressed throughout life? So we're going to find out why. All right, so the way I decided to tackle this is that we are now using a Cre-Lox system to be able to manipulate this TLE4 gene. Not sure how familiar you are with the Cre-Lox system. In general, you have this Cre system that is driving something else in this example. Okay, so here we're using the Cre to drive a Cre-dependent TD tomato, which is what you're going to see here in red. I actually was the one who characterized this transgenic mouse line tool. That's one of my papers from 2021. We're not talking about that. Anyway, so the first thing we found is that when we knock out that TLE4 gene, that FESEV2 is upregulated, and this is manipulating TLE4 at sort of a late gestational this is at E15.5, where mice are born at about E19 or E18, depending on whatever environmental circumstances. Anyway, all right, so we see that now cells are expressing this master regulator of subcerebral projection neuron identity. Great. Okay, we also see that when we when we knock out TLE4, markers for layer 6 are also downregulated. So layer 5 markers are upregulated, layer 6 markers are downregulated. So that's sort of a shift in cell type identity. All right. Well, we wanted to know, okay, is it changing the axon projection identity when I'm manipulating these cells in like these late gestational stages? And overall, we see that it is. What's going on here is that I'm injecting this retrograde tracer into the brainstem. This is using a surgical technique where I'm using ultrasound guided surgery to target the brainstem. So I'm injecting this tracer into the brain, and it's being uptaken into the axons of these cells. And overall, what we see is that now cells in the deeper layers of the cortex are projecting into the brainstem. So it's pretty cool stuff. All right, so like, okay, well, this is sort of fun, but what is going on at later stages of life? Can I manipulate the identity of these cells at later stages of life? And so what for me was sort of an obvious choice was to target cells at this pruning stage, which is about postnatal day P14, at which synaptic pruning, and there's a lot of synaptic plasticity still happening at P14. It was a pretty good idea. It worked out really well. Anyway, so in the way that we're doing that is that instead of using that transgenic mouse line, we're actually injecting a virus into the brain that expresses the Cre. Okay, so the virus injected into the brain, this Cre virus, is driving the expression of TD tomato and also knocking out TLE4. And then another surgery to inject that retrograde tracer. So it was a multi-surgery sort of trip with a double transgenic mouse line. All right, so here you can see I'm injecting the Cre virus into the mouse brain. This is a coronal brain section. I'm targeting the motor cortex, M1. These are the cells that are primarily affected in ALS. And so what we're seeing here, so when I inject this Cre virus in the brain and I'm knocking out TLE4 and expressing TD tomato just as a marker to see where I am at, I can see that FESV2 is upregulated when I'm doing these manipulations at P14. So this is basically showing that at P14, I can change the molecular identity of the cell. So at the transcriptome and genomic level, now cells are expressing these layer 5 subcerebral projection markers. Okay, well, you can, but what does that do downstream? Oh, yeah, okay, that's fine. Anyway, so then, but is it changing the axon projection at these later stages of life? Broadly, the answer is yes. This is a huge, huge deal. We're able to manipulate the axon projection of these cells at postnatal day P14. This took years of controls to confirm, and being grants rejected with reviewers saying what you're saying isn't possible. I mean, we couldn't have done it any more blind at this point. Blank viruses, mice I didn't know the genetics of that people would tell me later, all kinds of blind experiments to show that, in fact, we were able to reprogram the axon of these cells.
How long does that take, wait?
So we found, I mean, this change is happening over the course of about 30 days. So when we did it at 10 days to see, so we did the manipulation at P14 and then looked at P25, not a lot was going on. But when we looked at P45, P50, that's where the real changes were now starting to be sort of obvious. But those earlier experiments that I showed of reprogramming, like at embryonic stages or whatever, actually we have a whole P6 thing that I'm not showing anything of. Those changes could happen in like 6 or 7 days. So it sort of goes to show that the older the brain is, it can still have these big plastic changes. They just take longer to happen. That's one of the observations here. All right. So then we did a little bit of transcriptomic stuff. So what are some of, I'm just going to go back. So you're going to let me go back. Right. So what are some of the genetic elements driving this whole thing? So sure, we're manipulating TLE4. We're seeing this downstream thing of changes in axon connectivity. But what are all the genes in the middle? All right. So the way we went about this, and this is a sort of a cool thing to learn that is now sadly mostly obsolete, but it was pretty cool at the time, is that we're using laser capture microscopy to carve out layer 6 versus layer 5 of the cortex. This took a year to master, and it's really a cool technique. I just like to do things live now. Like the cells are alive in my hands versus this fixed system, but this is a big pathology approach, right? So we're carving out layer 6 versus layer 5 at different time points. Anyway, so here you can see what this looks like. This is that CYT6 Cree. So these would be the cells that are manipulated. And we also carved out layer 5 to be like that control of like, this is what layer 5 cells look genetically. Then these are control versus manipulated layer 6 cells. All right, so in a broad strokes, what we saw was that these layer 5 genes, these subcerebral projection neuron genes, are upregulated. So this is in let's see what this comparison is of. Okay, so this is in a layer 6, a control layer 6 versus a layer 6 that's mutated for TLE4. All right, so we're seeing that these layer 5 genes are in fact upregulated and the layer 6 genes are downregulated. Great. So we used this a couple of resources to define these, like, oh, how did we know that these genes were layer 6 versus layer 5? Well, people had figured that out, and I just sort of overlapped, just sort of combined the data. All right, and then a lot of these genes are in fact sort of developmental genes. So it's sort of reactivating developmental pathways. And a lot of these pathways are like signaling pathways, synaptic transmission pathways, cell morphogenesis, cell projection organization. So we have a lot of these sort of projection related pathways that were being activated. So that was a pretty good sign that these genes were regulating these pathways. All right, so then I wanted to understand a little bit more of, well, these are the genes that TLE4 is most likely directly regulating. Okay, so again, we wanted to understand the link between this TLE4 manipulation and is dendrite being affected, is axon being affected, is sort of cell identity, in what way is cell identity being affected? That was my big question here, right? All right, so let's see, okay, right. So these genes are regulating the overall expression. So if you take these specific genes that TLE4 is directly binding to, you can identify that these sort of clouds of genes are being regulated by this one or by this one. So that looks a little bit like this. We have that TLE4 is directly regulating MEF2C, and if MEF2C is then downstream regulating all of these guys. Now we know if these genes are cerebral, are corticothalamic or subcerebral projection, or what they sort of are. We know where they're supposed to be expressed. And so then now we have this multi-layer situation where I can identify, well, hey, what is this pathway regulating? Cell size, how's the cell myelinated, which is sort of a big deal in the projection, the synaptic organization. And so what we found is that there's some specific genes here that regulate dendrite development. So that was a big deal. We're now finding the pathways that TLE4, how TLE4 in a pathway type situation is regulating things like axon development versus dendrite development. All right. So broadly, we found, and sorry that this is so ugly, I haven't really figured out how to make it really beautiful. It's taken like dozens of iterations just to get here. Anyway, we find that a lot of genes and a lot of pathways are involved in axon development. Cool. But what was, to me, is really interesting is, why is dendrite development just regulated by a couple of these genes? Sort of an interesting, so we're, planning, oh, well, how are we going to manipulate these pathways to see what happens to the dendrite? Anyway, oh, here they are highlighted. Oh, I'd already done that. Great. All right. So, so we've talked a lot about cell identity. And I commented just in the very beginning how cell identity defines dendrites, but I've barely talked about dendrites. Why? Well, because we're going to talk about them now. Anyway, so layer 5 versus layer 6 cortical neuron dendrites. You see these neurons in layer 5 and they project these very large, very beautiful dendrites to layer 1. And these are thought to be critical in right now like there's this whole hypothesis of how these are like fundamental for cognition, for example, like particularly these apical tufts up here, thought to be critical for cognition. The cortex and how the cortex has evolved during millions of years is actually from this in to out system. So these cells are evolutionarily older than these cells, and cells in layer 1 are like the newest cells in both development and in evolution. So whatever is up here is more thought to be more advanced. Anyway, so how do we measure this? Like, oh, we can take a snapshot and see it. That's great. But how do we quantify this? We're doing shoal analysis to then look at how these dendritic arbors change, right? You just make these concentric rings around the dendrites and quantify their bifurcations. All right. So the way I went about analyzing sort of dendritic arbor is that I'm injecting our that Cree virus into a TLE4 knockout, to knock out TLE4 and then overexpress C tomato. But I'm injecting a Cree-dependent GFP virus in the brainstem. Okay, so I wanted to be able to isolate the dendrites of the neurons that were now reprogramming their axon. Sort of, to do dendrite reconstructions, you need sort of a sparse labeling. All right, this looks a little bit like this. Oh, all right. So, yeah, so now you can sort of see these dendrites of these neurons that are lit up by this Cree-dependent GFP. All right. Okay, so this is this retrograde GFP. You can see here, this is in a control situation where we have neurons in layer 5A, neurons in layer 5B. We can then come in and semi-automated do these neurolucida reconstructions using like a $30,000 software. It's really expensive. And so yeah, so then when we do the mutation, what we see is that now we have neurons in the deeper layers of the cortex projecting into layer 1. This isn't the most recent data set, but overall it shows that we have neurons at least tending to project towards layer 1, which is sort of a big deal. Okay. All right. So overall, cortical thalamic neuron identity is plastic, and reprogramming can happen at different life stages. Axon connectivity can be altered up to P14, and dendritic arbor can also be altered to at least P14. All right. So thanks. This is just sort of the first part of this. And yeah, this is it. We're just going to stop if there's any questions. Is there a question in the chat? Oh.
How does the function of those now compare with control, the regular state, right? Like, are these being incorporated into functionally important pathways? How does behavior look, et cetera, right? So
Totally. If I had another year, that's what I would have done. And there is someone else was dedicated to doing that, and it was because our reprogramming approach is not a consistent approach. It's like a stereotaxic surgery. Like, let's see what percent of neurons actually reprogram.
Oh yeah.
We found that it was anywhere between 0.2 and 3%. So that's a couple of fold change differences. So yeah. That was one issue. A lot of resources have gone into trying to figure out if there are, in fact, synaptic and physiological changes.
Yeah.
We'll also see what the reviewers say if they force us to figure it out.
Yeah.
Then we'll have to do it.
Yeah.
Yeah. That's where you submit and it takes 2 years to publish.
Exactly. Yeah. Interesting. That's really, really cool data, though. Any other questions?
I'm sorry if I sort of skipped over the genetic tools and how they work. I would really just love to talk about that one day, just how beautiful these techniques are and how, you know, we can use all these virus cocktails with transgenic mice. And so it's the mix of fancy viruses with constructs with fancy mice and fancy surgeries.
Yes, and we'll talk a bit about that on Thursday, because this is real, like, one, it's a ton of work. It's a ton of work. It's a ton of work trying to characterize these tools on their own, but then the combination is, I mean, it's pretty insane. Like, to have, you know, 3 or 4 of these approaches being linked in a single experiment is incredibly complex, and it's also not easy to do.
Yeah, I just kind of want to ask, how easy would it be to replicate this experiment later on?
So replicating it quite easily. That's the thing. A person with a large lab could do this very quickly. It's one of the reasons that we haven't made it publicly available, even in a preprint, because people will figure out how to do this. They have since created a number of TLE4 lines. So you sort of have to wonder about this, is that the efficiency at which we can reprogram those cells varies.
Yeah.
But the success, the binomial success of yes or no, doesn't really change. It's always a number of, is it 2 or 3 cells that we see, or is it like 150 cells? So it's sort of, it varies like that. There's sort of a epigenetic stochasticity that might be at play as well. How can we make that more consistent? That was sort of a biotech drug idea that came to mind. Is like, well, how can I make this more efficient using drugs applied to the brain, you know, or some kind of thing? Got some pretty good ideas of how to do that, like opening up the genome to make everything more accessible for these transcription factors to take function. So it can be easily replicated, but the efficiency is sort of, you just have to know about that.
Yeah.
And
I had one more.
Yeah,
Sorry.
go for it.
On your last slide or two, in terms of the terminals on those dendrites, yeah, so here, and yet, like, is the lack of extensive branching of those terminals, is that just due to like time of expression, or is that a real, you know, is that not happening?
We didn't, so at least in this data set, while they project to layer 1, that really complicated apical tuft, it's not forming in a lot of detail is what we're seeing. But also sometimes it's the expression. So we're going to see, like, you know, you need some statistics to show that. We'll see.
Okay.
We'll see how this all looks. It's not looking great for the apical tuft, but even just projecting to layer 1 is a big deal.
Oh yeah, no, that's huge. Cool. Yeah.
I'm just curious, so I think about like stem cells and how once they're like differentiated, like there's no like going back. And so with neurons, like how, am I understanding this right, that they're differentiating and then changing? Is that correct? Or is it like the early stages that you're kind of, yeah.
I love this. Thank you. Wow. My boss would hire you on the spot. Okay. Just right there. I'm sorry, I just couldn't contain myself with you asking this question. So there's something, there's a principle of molecular biology that's called the Waddington landscape. I don't know if any of you have heard about it. It's sort of its own thing where cells will sort of tend to go in a direction, right? It's sort of a ball rolling down a hill, and through development, they are defined, right? And then going in reverse is really difficult, right? So here, you know, instead of, for example, you commented about, maybe you said something about stem cells, is that when you take a cell back to a progenitor stage, like a stem cell, like a multipotent stem cell versus a pluripotent stem cell, right? That might sound a little bit familiar. Well, how do you then redirect it to a specific cell type, right? So you know, so going in reverse of that Waddington landscape, of that hill of development, it is so significant that someone won a Nobel Prize for it. Right? Anyway, what's really particular about our approach is that we are converting one cell type into a very specific other cell type, right? When you do your stem cell pluripotent experiments, it's like, well, here's this homogeneous group of stem cells that we've all like converted back into stem cells, and then they're going to be this other soup of other stuff, right? Sort of a soup of different cell types, depending on your cocktail of transcription factors and morphogens. Here with one gene, we're doing all of this. It will be someone else's job to find that second gene that will make the apical tuft go like really flower up, right? So anyway, yeah, Waddington landscape, big deal, big deal.
Thank you.
Yeah, great question.
I have a follow-up question as well, if that's okay. I'm just curious, so I'm guessing like this research is done just to understand like how the brain is working, but if like you were to zoom out, is there like a broader goal? Like is it to be able to kind of alter like an older brain and bring this into that? Or like I'm just curious about that, like future.
Totally. So that F99 that I got was how to, and that was sent, that grant is to the NINDS, which is this National Institute for Neurological Disorders and Stroke. So this research was in part funded by mechanisms to find cures for ALS, right? So there's different types of ALS, and the upper motor ALS, these are exactly the neurons that are affected in upper motor ALS that degenerate during that neurological disorder. So if we can, so in ALS, these cells will die. Can we now convert these layer 6 neurons back into these cells? These are cells that are regulating all sorts of functions in the, you know, I mentioned these subcerebral projection neurons are projecting into the brainstem and all kinds of bodily functions. If you start to kill those cells, that's a form of ALS that's happening.
Thank you.
I've been asked, I'll throw in a, you know, there's a bioethical question that sort of comes up. And just to, you know, keep in mind, sure, I'm making this, you know, cool stuff to be able to rejuvenate the brain and help people with ALS and discover these pathways. You could, in a very anti-ethical approach, use this to reprogram human cells in a person who does not want to be reprogrammed. That is a possible use of this technology. So it sort of comes into play of like, well, sure, I'm making all these cool discoveries. What are the potential risky applications of the thing that I'm making? And knowing these, that's sort of a scientist's responsibility. You know, just like CRISPR, right? It can cure every type of cancer, great, but then it can do a lot of harm. So what are the ethical implications of your research are not always obvious.
Any other questions about the first part?
What things would it do? What unethical things would it do to the person?
We don't know. We don't know. Okay, so when we are when you're switching the cell type from one cell to another, you're changing its synaptic connectivity. That is changing its cognitive processing, its memories, its memories that have been stored throughout its life in that cell. Now you've switched its identity. Do all of the synapses collapse and then it builds new synapses? We don't know. There's a group who wants to work with us to see the live imaging, like, oh, does it just like subtly change the memory that existed? Or does it blanket it out and create a new one? We don't know. So it's the thing about, you don't know what your technology can do, but you know it can be used for harm, right? And bioethical committees in universities keep, like watchdogs, keep an eye out for this kind of thing. So they have an eye out for us, of course, because look at this, you know. But we reached out to them and said, hey, we're reaching out to you. Look at this. We know what we're doing. Just know that we know.
So, sorry if I'm like getting this wrong, but I'm just wondering, so like if you're saying that like you can like change a cell type, with the cells that are dying for like ALS, could you change like a different cell type into the ones that are dying to like help like treat the ALS? Is that
Yeah,
like,
that's exactly what we're doing.
okay, I just wanted to make sure I was like getting that right.
I take clarification questions, of course. That's one of my go-to talk questions. Like, did you say this thing?
Thank you.
Are you focusing specifically on the sensory motor integration part of it? Or
I'm sorry, can you repeat that?
are you focusing mostly on the sensory motor integration, I think you're saying?
Yes,
Thalamus?
yes. I had to choose one brain region. That's it. You know, when my boss gave me the projects, and we're like, oh yeah, like what's the difference between sensory cortex versus motor cortex versus M1 and M2, S1, S2? Oh, that's insane. I mean, so we have some preliminary data about differences in the success rate of reprogramming depending on the brain region. We just don't have a powerful N, but we do see a little bit of a subtle difference, and there is an evolutionary explanation for this.
Cool.
So yeah, a guy in the lab is now working on the prefrontal cortex, right? So it's sort of, we'll see what happens.
Have you guys done anything yet in hippocampus?
It's
Yes.
sort of some guys doing something. It's a little bit of a stretch. TLE4 isn't expressed in the hippocampus, but then the prefrontal cortex, thalamus, hippocampus axes of spatial memory that we'll talk about now is, so prefrontal cortex, thalamus, hippocampus, all this interconnection of spatial memory formation. He's manipulating TLE4 and then looking in the hippocampus to see sort of like, well, what's going on later. Man, is that data ever dirty? But you know, it takes a while to see, well, what is the best way to look at a problem and to look at the data. So
Sorry, man,
You mentioned that you use like a laser technique on live cells. Could you explain that a little bit more and how that works?
So it's not on live. I will talk about my laser stuff on live cells in a minute, but you mean the laser capture microscopy? This is on fixed tissue.
Okay.
But it is, I mean, the cells were alive a couple of minutes before, right? So you sort of have to very quickly do this. Something like within an hour and a half to a couple of minutes, you know, flash fix, mount the tissue, do this collection. I really thought that we should have done sort of a single cell sequencing approach, but if you were to take this whole brain chunk and then isolate the single cells, you don't know where the cells were. We are changing the layer 6 into layer 5 cells. So if you mash them all together, what is what, right? We've seen these data sets, and man, are they ever a hodgepodge of all kinds of crazy statistics to figure out what they did. So.
Thank you.
Sorry, Aisha.
No, it's okay. I'm just wondering, like, how would you go about kind of like figuring out how this technique would work on like live humans? Like, is there any way to use like machine learning models or like computational models to kind of better understand?
Yeah, totally.
Yeah,
totally.
or need a lot more data.
Okay, so there is two ways that really come to mind. It's that I'm an experimentalist, right? So when it comes to, I do have a little bit of modeling experience, but in the end, when you manipulate tissue directly, that's where things start to, the model is to prove the experiment, right? Or guide experimental decisions, but it doesn't, anyway. So one way is to use cerebral organoids. I don't know if you guys are familiar with this. This is like a technology of your generation of taking cell samples from patients and humans and then converting them back into multipotent cells, and now they've started to divide and to create a little brain in a dish. Any of you familiar with this? A little bit? Okay, so cerebral organoids, huge deal. The research is very slow given that they grow at like a human pace. They take, months and months to develop. And they're really hard to grow. Human patient derived organoids. Our biggest competitor is now one of the world experts in this, and we're just like, thank goodness that she shifted directions, because we needed a little time to catch up, honestly, and just thank goodness. So another way is using, so for example, if you go in to have, if you have intractable epilepsy, you might have that epileptic part of your brain resectioned, right? If that is the elliptogenic, if that is the site in your brain that is causing the seizures, we can just pull out that part of your brain, that tissue section, which they've, used electrical stimulators to identify, oh, well, what is the part of the brain creating the seizures? Neurosurgeon stuff. Anyway, they'll take out this brain section. So what we can do is keep that brain section alive for up to weeks in a cerebral spinal fluid bath with a couple of hormones mixed in there, and do some of these manipulations and just see what is going on. I have a little bit of that I can show you that's in these slides. Anyway, does that answer your question?
Yeah, thank you.
Well, how much time have we got for this?
Until 2:30.
Until 2:30. Okay, so we have another half hour. Sorry, I'm on a different time. Okay, so I'm gonna sort of move quickly through my next part, and then we'll do this again. All right. Oh man, that's so much stuff. Okay, so working in fixed tissue was great for my PhD, but man, I'm a good surgeon, and I said, you know what? I need to work in live animals. I need to do stuff with in vivo situation. What's going on with this tissue when it's bubbling on a dish and I can manipulate it? What is happening live? Okay, so I decided to join the Simon Chamberlain lab at UPenn. I'm his first postdoc, and he had a $2 million microscope that no one was using, and I said, I'm going to use that microscope. So what you see on the left is a neuron that we are recording with a patch pipette, patch clamp electrophysiology. We filled it with dye, and we're looking at both electrophysiological and physiological things that are happening in the dendritic arbor. This has been pretty cool last couple of months. Anyway, so the goal of this project, it's like sort of the entry-level project of this lab, which is, we make it sound, it's the entry-level project, but it is super complicated. Anyway, how does dendritic inhibition manipulate these plateau potentials in CA1 pyramidal neurons? So I'll just talk a little bit about what these plateau potentials are. But so this is a totally different project. So we're going to gear up. All right. So as a mouse is running around in sort of this open maze, it create sort of a spatial map. You have a spatial map of where you are located right now, the spatial cognitive map. And if you implant an electrode in, like, to record a couple of cells, you'll see that they become sort of tuned to specific locations of the sort of open field. Okay, so now these cells have become tuned to that spatial location, and this is doing recordings in the hippocampus. People have known this since the 70s. All right, so the hippocampus is really the site for this place field formation, and these CA1 pyramidal cells, the cells that are located here in the CA1, are the true masters of that place field. Okay, a little bit about the circuit, we won't go into that. Anyway, so what people have found, and this is just one of these cells, there's a lot of forms of learning. 10 scientists in a room will say 10 different things about learning, and they're all correct. But in this form of learning, that's sort of this one-shot learning, where a mouse is running on a treadmill. The treadmill has a couple of different sort of markers, so it knows where it is on the treadmill. You put it on for the first time, it's running on the treadmill. But what'll happen is that after about 10 laps, the mouse will now, oh, this is how the treadmill works. Hey, the glued little things here, that is this location. Okay, so that feeling that you get, oh, I know where I am, that is a signal in your brain that is happening in place cells. Okay, so that is a large calcium wave that is happening in these CA1 pyramidal cells. It's called a plateau potential. Primarily calcium I'm not talking about the mechanisms of it, but just know that it is very different from single spikes or burst spikes of action potentials. It is a large calcium wave. And you've seen in your basic biology, neurobiology classes, how calcium is this critical secondary messenger, just signaling all forms of like memory and plasticity, right? So large calcium wave, let's trigger this plasticity, let's create this memory, and also signal we know where we are. Great. So the way, this is a little bit about this one-shot learning, and it is that sort of exemplifying this element that we're recording from CA1 pyramidal cells, we're stimulating these other cells that synapse onto the CA1 pyramidal cells in this radiata layer. And what you see is that this is the first stimulation wave, and after a plateau potential, if you force the cell to have one of these plateau potentials, now this synapse is potentiated, right? So it's sort of the power of this calcium wave in potentiating this synaptic connection. All right, so, but what is it about these plateau potentials? So reason I joined the lab is that I knew that these plateau potentials were generated in the apical dendrites of the cells. So you might have action potentials which are generated axosomatically, this kind of thing. Well, plateau potentials versus action potentials, you have that these plateau potentials are regenerative electrical events happening at the dendrites. I found that that's pretty cool. Instead of action potentials, you have plateau potentials, and instead of in somas and in axons, you have that in dendrites. Totally wild. Anyway, but what was interesting is that these plateau potentials are happening in the dendrites in this dendritic region, which is the same place that the somatostatin interneuron, an inhibitory neuron, is also synapsing. So it sort of led us, you know, and people have found that somatostatin interneurons and inhibition in general has like key roles during learning and memory. So it sort of led to the question of how are these somatostatin interneurons regulating these plateau potentials? I don't have this slide here, but the thing about these plateau potentials is that people argue that they're very difficult to generate. Oh, you know, they are a sort of a two-hit signal that, you know, when you so when you've learned something that is like an associative memory between two things, that's when this plateau potential happens. Really, when you put a brain slice on a dish and you're recording from that cell, plateau potentials are really easy to generate. You can generate them by just messing with the temperature. You can do a lot of things to generate plateau potentials. But You walking into a room, you don't want to walk into a place you've never been in and say, oh, I know where I am. Oh, I know exactly where I'm standing. You do not want to create an erroneous place field. That is a very bad thing to do. That could, in a bird or a mouse, that would kill you in the environment. So how do we control the generation of these place fields via the control of the plateau potential? That's the question. All right. So what we found, what we're doing here, okay. So what we're doing here is that we're generating a plateau potential. We're recording with these cesium pipettes in the pyramidal cell. You see a plateau potential here. This is a calcium wave. And then we're using optogenetics to stimulate the somatostatin interneuron. What we find is that when we stimulate the cell with this, I don't know if you guys know what channel rhodopsin is, but it's basically activating the cell. When we activate the somatostatin interneuron, it's able to stop the plateau potential in its tracks. No one has seen this. This is sort of like a total serendipitous, really crazy finding. Things are rarely like this clear cut. Anyway, all right. So what we found is that it was sort of an all or nothing element where, you know, you get this bout of inhibition if you're not successful at terminating the plateau, like a unsuccessful termination, the plateau potential just comes back. But if the inhibition is strong enough, it will shut down that plateau and just signal to the brain, hey, don't make a memory of this. This is not what you want to make the memory of. All right.
So
Yep.
I have a quick thing. How exactly are you measuring these, or like what, like, or spatially speaking, like where you're technically speaking, how are you isolating this potential? How is it different from like other, you know, other
Like
sensors?
other. I'm sorry. So how am I isolating the plateau potential?
Yeah, like how exactly is this defined, or how exactly is it measured?
Totally. Okay, so right now it is defined, it is primarily a voltage gated calcium channel mechanism, right? So with enough voltage and calcium that is activated from the action potentials, you will then, so that's why the plateau potential is generally before this action potential, right? So the backpropagating action potential activates the plateau potential, but single action potentials are not really able to do it. You need more mechanisms at play so you need more things at play.
And I know you said, so is this calcium largely coming from outside the cell, or are these also internal stores that are being released, or not, you know?
Oh, that is a good question because it is one that we have not answered yet.
Okay.
Thank you. I'll bring that to my boss. Like, oh, someone asked that question that we hope no one would. Nice.
Well, that's because I'm interested in, you know, internal stores and like, yeah. So this is kind of stuff that I'm interested in.
Totally. Oh man. Yeah, that's more drugs to apply to the brain and seeing what's happening. We think it's mostly external calcium, but you know, it said there are also different, so, and yeah, I don't know our time, but this is one type of calcium conductance that's going on here. Well, it's two, but you know, we'll just say it's one for this here. This is one type of calcium conductance, but then this here, this little wave here is a different calcium thing that's going on. And man, this sucker, no one knows what it is. We have gone through, you know, the 50 years of literature. People have no clue of what this is. So are we gonna? I
mean, it's like a longer live, you know, it has slower kinetics. Interesting.
Totally. People have like, they just observe it and they're like, oh, we see that thing that no one knows what it is. You know.
I'm curious, how is it different from, is it called like the refractory period or whatever, like after an action potential? This is different from that?
If we were to call it anything as in like a refractory calcium response, we just know it's calcium. it's some sort of calcium transient. There are over, man, what am I gonna guess? Like 45 different types of calcium channels. Something like that. It's really, and then you have not just one calcium channel, but you can have the combinations. We don't know. We don't know, you know. Some biophysical person was explaining this to us in a language that I could personally just not understand of like the biophysics of this.
No drug yet has blocked this. Yeah.
Oh, well, if you do like the everything calcium,
Yeah,
like
yeah.
if you block just all of the, like if you
Yeah.
do the super cocktail, it's gone, right?
Yeah.
But then you don't know, like, oh, did you block one of the two things? Like, did you block the gate of it? But it's still there, but you just blocked like the first step of it. We don't even know. We don't even know. Anyway, so broadly, we are able to terminate these large plateau potentials, right? So signaling, hey, do not form this place field. I'm just gonna make sure that I have this in the right order. Man, I knew that I would be cutting it short. Okay. I cut a lot of the I cut a lot of the talk to be able to have this here today. Okay, so when we're recording from the soma, we see that the electrical properties of the cell, right? Of this is what you're seeing here is this calcium transient, and that is now reflecting as a large change in voltage. This is all voltage that we are recording from the soma, right? Great. Good for you guys. You're seeing that you can terminate this calcium plateau that, you know, this voltage plateau that you know is calcium, and you're doing it at the soma. Great. Not great. What is going on at the dendrites, and what is going on with the calcium at the dendrites? Okay, so here we're doing a kind of 2-photon imaging that's called random access 2-photon imaging, where I am now filling the cell with a red dye and using a green calcium dye to see the changes in the calcium. Okay, so this is a calcium buffer. It binds to calcium and fluoresces. Okay. All right, so we can fill the cell. I already commented about that. We fill the cell with the Alexa dye, and now we can target different regions of the dendrite and record their calcium responses. So what you're seeing here in the darker traces are, so this is sort of in this primary dendrite region right here, and then this is oblique dendrites projecting out here. The darker color is the control, and as the color becomes sort of lighter, it is earlier and earlier terminations. So anyway, what we're seeing is that we're able to grade the calcium response when we are terminating plateaus at the dendrite. What's the point of this, right? And it goes against what I said in the beginning, that there's like, while the plateau in itself, the electric element is sort of this all or nothing mechanism, the calcium in itself is sort of a graded response. So we're sort of still untangling what this means. Like the next paper is really to look at this in vivo with the mice running around and all kinds of cool stuff. That's the next step. But overall, different concentrations of calcium have different effects, right? And so what's happening now is that we're converting this all or none signal. While it's a voltage all or none signal, it is a graded calcium response. So pretty cool. It was thought previously that the calcium was also an all or none, right? They're going to ask us to test more stuff because we're going against dogma. So we'll see what happens. Another experience of going against dogma. We'll see what happens. Anyway, oh yeah, so I had a little bit of the quantification here. So, you know, we're terminating that voltage at different times, and we're seeing this graded response. Oh, great, I did put it in. Cool, okay. So my PI is a new PI, and like young PIs do, they sort of come together and make their PI click. And he met this guy, Sean Sorrell's, like really one of the nicest PIs I've ever met. And says, hey, you know, I get this human patient tissue, exactly what I was talking about, the resectioned seizure tissue. Hey, I also get hippocampus. People with whose that the hippocampus, or one of the two sides of the hippocampus, is the center for that epileptic generation to then cause the seizures. Anyway, so this neurosurgeon is the one who would supply the hippocampal tissue to him. And so now we were getting it supplied to us. So basically people, you know, they remove these pieces of hippocampus from their brain. We generate acute slices, and here they are, just live human brain tissue floating in this cerebral spinal fluid. And then we go and do our electrophysiological recordings. So broadly, and you know, there's sort of some overall, you know, what is going on in terms of, you know, if you look at a pyramidal cell, at a CA1 pyramidal cell in humans versus mice, these are, I mean, these are orders of magnitude different in size. But what does that mean in terms of sort of place field formation? We can do these EEG recordings and seeing how people are interacting with their environment, but at the physiological level of the neuron and the dendrite, we have no idea. Anyway, people had not seen plateau potentials in humans. We see plateau potentials in humans. Huge, huge deal. And then we're also able to terminate those plateaus in humans with using just voltage, injections of voltage. You see these regenerations. We don't understand why these regenerations are happening really. They also happen in mice, but they happen in mice in very specific contexts. In humans, you see like multiple plateaus happening, right? We're able to stop one plateau, but then their regenerations are still there. What does that mean for memory? Instead of just having a calcium wave, now you have like a Morse code of calcium. What does that even mean?
And Lee, just a quick question. How do you, are these neurons that are not unhealthy or not, you know, like, are these ones that are normally involved in generating the, you know, like how relevant is this to?
We get this every time anyone talks about this, we get this. Well, they're not dead.
Yeah, they're not dead, but.
They're not dead.
Yeah.
We have received different cases from different neurological disorders.
Yeah.
And we see the phenomena in all of them.
Okay.
But the repeating calcium waves, we don't have enough statistical power to understand what's going on. Maybe that's one of the sources of the seizure.
Yeah.
We don't know.
Yeah.
But just seeing the plateaus in humans and seeing we can terminate them.
Yeah. Well, that's
pretty cool. There's sort of some manipulations. We're trying to think of how to see these in mice, and then like what happens if we generate these repetitive things in mice, right? Like what happens? Is that it? Oh yeah, that's it.
really cool. Questions?
I mean, I kind of had the same question you had about like if you're getting these samples from neurosurgeons or from people who are like it's like sections of the brain that need to be resected for the brain to function normally, then how, I guess, do you think that if you did the same kind of experiment on like a normal, like regular functioning part of the brain, do you think these results would be replicated, or do you think it would be slightly different? Do you think we just don't have enough data to know yet?
We don't have enough data to know yet. We are now, for some reason, a lot of these surgeries happen during the summer. In like 6 months, we've gotten 3, but now in the summer, we're getting 6. So we don't know why the surgeons are doing this, but really the statistical power is the way that we're going to go about this, right? So the surgeon is a pediatric surgeon specialized in epilepsy, but he does a number of other things. So in the end, we'll see what's happening. This data will take either months to submit something medium or years to generate something really crazy. We'll see what this all means. We actually got since I built this slide, we got another case with even another like weird thing that no one had ever seen before. So these human plateaus are they the thing that are, you can generate your spatial maps of any place in the world, of the planet, right? These people who look at Google Maps and it's just like they know it by heart. What does that, could you do? What mice? We don't think mice can do that, right? So, we'll find out about this, about your question. Is that a seizure? Are we seeing a seizure in a cell? Seizures don't really look like that in a cell. They're more spiky and crazy and more out of control, but
how common is this preparation, like, in terms of, doing, these kinds of recordings, how common is this kind of data in your field or in the broader field?
super rare.
Okay, I was gonna say, like, I don't think I read a paper that.
Very few. I'm gonna say maybe
Like
100 labs in the world, something like this. And it's like they're getting a human brain every, twice a year or something like this. I mean, but if it's like the Allen Brain Institute, they have all of these places in Seattle, everyone's sending them brain samples. That's different. I mean, our statistical approaches are totally based on how the Allen Institute does their analysis, which I imposed on our team because we need a standard. What is the standard? Oh, who's creating it? Oh, well, then we'll use their standard. The opportunity to do this, I came from doing aplesia marine animal brain science, and now I'm taking human samples and testing them, right? This is way beyond what I imagined originally, but it is really cool. My intention is to go back to marine animals at some point. It's sort of one of my, I sort of dream about this, but man,
It's tough.
it's tough when you get an opportunity like a once in a lifetime type of opportunity.
And just a quick follow-up and that gets, have you done any fun, like, have you stained these with any, like, have you done anything wild with these neurons at all? Like, if I had access to this, I'd be thinking, like, what do I really want to see in like a human neuron? A live human neuron. I tried some live, have you done any, anything that's not even tied to one of these experiments, just for fun?
So yeah. So one of the things we're trying, the Chamberlain lab is really big into identifying new interneuron subtypes, right? The whole inhibitory cells thing, right? Well, there are dozens upon dozens of different types of inhibitory neurons. And so, we take a lot, like, we have 3 projects that we do with this human tissue. And when this is happening, we might be working for, 16 hours straight, like until the brain is like not functioning anymore. We are working nonstop. So we have a couple of projects that are involved in identifying different cell types. Yeah, so they're staining for all kinds of crazy stuff.
Yeah, nice. That is really,
Also, the morphology of these cells, it's a whole story. They have a whole thing on these LAMP1. They're actually layer 1 cells. I was talking about how the neurons in the upper layers of the cortex are these cognitively highly advanced. Well, they're studying, what is the diversity of those hyper advanced cells that only reside in layer 1?
That's really, really cool. Yes.
Totally.
Since the surgeon's a pediatric surgeon, are these children's brains or adult brains?
These are mostly between 9 months and about 16 years old that we're getting these tissues. So these are kids, these are kids, and when we get these samples, you might see me relax today. you know, and I'm pretty good at being chill with my students, but this is, you know, this is, how do you say this? The value, the human life that you're working with now, we don't accept any mistakes when you're in working with us on those days. It's not acceptable. So, you know, these are really rare opportunities, right? To do science with someone else's, you know, this sample, sure, it might have been tossed away or used for something else, but still, we have to value it.
The reason why it might be, you see more things in the summertime, our kids are off school in the summertime, so they wanna take.
Oh, thank you for thinking of an answer to that. Wow. Man,
Yeah.
we just didn't understand what was going on.
Yeah.
Yeah, to recover and go through
Recovery
therapy.
time,
Yeah,
yeah.
totally. We got one during Christmas.
Yeah, so you're off on break for Christmas.
Totally. So that, man, so looks like my trip to France is gonna be messed up for this summer. We'll see.
Wow, that's really cool though. Really cool stuff. Any other questions for Dr. Lee or any training questions? Oh, well, thank you so much, Lee. That was really cool stuff once again. Talks are always illuminating and it's always fun to see what science you're working on. This is really cool. So it's gonna be a fun discussion on Thursday for sure.
Totally.
Good stuff, man. Really good stuff. I look forward to those papers, man.